My name is Joshun Polaru, and my project's title is Identifying Manipulators of Ambulation and Implications for Parkinson's Disease. Prior to starting this project, I was introduced to a lab that aligned heavily with my research interests. They had just done a high-throughput screen of several compounds, and my research goal was to identify promising manipulators and verify their effect on Huntington yeast protein E, or HYPE. Ambulation is the attachment of an adenosine monophosphate, as shown here, to a substrate. There are two important constructs of HYPE, wild-type HYPE and E234G HYPE. E234G HYPE is an overactive amylator to the point of possibly endangering cells, while wild-type HYPE is intrinsically sterically hindered and can therefore only weakly deamylate certain targets or weakly amylate. HYPE amylates two important substrates, alpha-synuclein and BIP. Alpha-synuclein is a protein that has been heavily implicated in the progression of symptoms in Parkinson's disease. Its oligomerization and then aggregation into fibrils and Lewy bodies is a common hallmark of symptoms. However, when alpha-synuclein is amylated, as shown here, it can no longer properly oligomerize, thus alleviating the cytotoxic phenotypes. Hence, one reason to find an activator of wild-type HYPE or an inhibitor of EG HYPE would be to reduce these cytotoxic phenotypes without endangering the cell. BIP is a protein involved in the unfolded protein response, or UPR. BIP's activity is mediated by HYPE. If HYPE amylates BIP, BIP will remain inactive, and if HYPE deamylates BIP, BIP will become active and refold misfolded proteins. One consequence of the UPR is apoptosis, as shown here. Some cancers, as a result, suppress the apoptotic branch of the UPR so as to avoid cell death. Thus, manipulators of HYPE could then increase BIP's activity, possibly leading to apoptosis of cancerous cells. Now, with these factors in mind, I designed several in vitro assays. The high-throughput screen that identified compounds operated on fluorescence polarization. When FLATP, the fluorescent construct of ATP that was used, was bound to some substrate, it could not depolarize light, and the amount of polarized light was then measured. The high-throughput screen also used a construct of HYPE with the first 102 residues from the N-terminus removed. I chose to use a construct of HYPE with only 45 residues removed from the N-terminus, because even though it is much harder to purify, it is much more physiologically relevant. I also directly measured amylation using a fluorescence imager to detect signal from FLATP, so that signal could not be confounded with anything else like a reduction in Brownian motion possibly could. The darker a band here on these gels, the more amylation has occurred. Since there may have been some error in pipetting, all data were normalized to the stained gel so as to avoid confounding. Activators 1 and 5 and inhibitors 1 and 2 function the best in this assay. I repeated these experiments for BIP and alpha-synuclein, and activators 1 and 5 and inhibitors 1 and 2 continued to perform consistently. I wanted to test a third activator in cells, so I chose activator 4 due to its greater success in BIP amylation. I did not choose activator 3 for use in cells because it appears to be an outlier due to its almost non-existent function earlier. As a result, I went forward with activators 1, 4, and 5 and inhibitors 1 and 2 in cells. I cultured glioblastoma multiforme cells in order to collect cytotoxicity data and perform an anti-adenosine monophosphate Western blot. Unfortunately, due to some delays in the arrival of necessary cells, I am currently still waiting on the results of the Western blot. But I was able to collect cytotoxicity data using a trypan blue live dead cell count. As shown here, live cells appear to be whitish because they have not taken up the dye, whereas dead cells are much darker because they have taken up the dye. After using a hemocytometer to obtain cell counts on each well and obtain cytotoxicity data, I normalized to the control since DMSO is not cytotoxic. Most compounds appear to not be cytotoxic, but activators 1 and 4 do. This could be because of the aforementioned effects on cancer cells, or just because the compounds are actually toxic. Several compounds showed promise as therapeutic targets for Parkinson's disease in vitro, and they could show promise in cells depending on the results of the Western blot. Further experiments could include checking the expression of commonly expressed genes in the UPR in order to determine whether it is apoptosis caused by the UPR or just cytotoxicity from the compounds that is killing the cells. Other experiments could include testing multiple compounds at once and examining synergistic and antagonistic effects of those compounds.